Hello, everyone, and a warm welcome from FSR. We'll begin our webinar shortly here, but please know that you are connected. Hello, everyone, and thank you so much for joining us this evening for FSR's latest grant awardee webinar entitled Discovering Pathogens in Neurosarcoidosis. I'm Tim Legensoff, FSR's Senior Research Manager. Before we begin today, I just wanted to share a few quick housekeeping items. This session is being recorded. All attendees will be muted throughout the presentation. There will be a Q&A at the end of the presentation, so please feel free to submit your questions in the chat, and our team will strive to bring them forward during the Q&A. Please note that all questions that are submitted can be viewed by all of our attendees today. Please do try to be as broad as possible when asking your questions in the chat. We know how unique sarcoidosis is, but it's important to note that our expert today is not able to provide any direct medical advice. I'm now happy to pass the webinar over to my colleague, Elise Hoover, FSR's Vice President of Research.
Hi, everyone. This is FSR's second grantee webinar. And so our goal is to provide funded researchers with the opportunity to share the results and impact of their work with a larger community. Thank you so much for joining us. And because FSR has funded a large number of projects, we look forward to many more of these webinars going forward. I am now excited to introduce Yvette Cozier, a key leader of our organization and a member of FSR's board of directors. She brings a deep commitment to advancing sarcoidosis research and empowering the communities that we serve. So please join me in welcoming Yvette, who will now share a few opening remarks to set the stage for today's webinar. Yvette, over to you.
Thank you, Elise. Good afternoon, good evening, and good morning. Thank you for joining us from across the country and around the world. On behalf of the FSR Board of Directors, it is my privilege to welcome you to today's grant awardee webinar, Discovering Pathogens in Neurosarcoidosis, featuring Dr. Paula Barreras. Today's turnout shows our shared commitment to understanding neurosarcoidosis and challenging and often overlooked form of the disease that can have profound impacts on quality of life and neurological functioning. To put today's presentation into context, this is FSR's 25th year. In the organization's history, we've supported over $8 million of research funding on over 50 projects. This has translated into leveraging over $10 million in follow-on funding from the NIH and other large funders. This is important for researchers to not only continue the work that they've embarked on, but to answer their specific research questions, and it ensures that they stay focused on sarcoidosis. You may or may not be aware that all of FSR's funding comes from private donations and corporate support. FSR does not receive any government funding. If you wish to support FSR or get involved in any way, please visit stopsarcoidosis.org to learn more. And now it is my pleasure to introduce today's speaker. Dr. Paula Barreras is an assistant professor of neurology and director of the Center of Excellence for Rare Neuroimmune Disorders at Cedars-Sinai Medical Center, which is a founding member of the FSR Global Sarcoidosis Clinic Alliance. Her research focuses on identifying predictors of outcomes in neurosarcoidosis and understanding the causes of sarcoidosis-associated small fiber neuropathy. Dr. Barreras was awarded an FSR fellowship grant which is designed to increase clinical and/or scientific engagement and commitment within the sarcoidosis field. To date, FSR has funded 13 fellowship awards for a total of nearly $2.5 million. Dr. Barreras serves as a model for FSR's goals for this program. She is an early career researcher who has secured a full-time academic position in sarcoidosis and continues to advance support of patients with sarcoidosis both in the clinic and through her research. I am delighted to turn it over now to Dr. Paula Barreras for her presentation.
Thank you so much for that introduction, and it's always exciting to hear about the amount of support that FSR has given to trainees, and like myself, I hope many in the future follow in this field. So now I'm gonna share my slides. Okay, so I'll be talking about pathogens in neurosarcoidosis. And I'll start by thanking FSR for their support for not only this specific project, but really for my training. The grant really allowed me to receive training from experts in neurosarcoidosis specifically and systemic sarcoidosis, and focus really my career in neurosarcoidosis moving forward. And the second disclosure I want to make is that everything that I'm showing here that was done as a result of this fellowship award was a team effort. So in research, it's always important to keep in mind that there's not a single person that can do a whole project. It was collaborative work, and under the guidance of my mentor, Dr. Carlos Pardo, who's at Johns Hopkins still. I want to start by talking about sarcoidosis in general. I know this audience is mixed, and we're all here because we're interested in sarcoidosis, but we don't really know yet the specific triggers or the specific cause of sarcoidosis. The belief is that something in the environment, perhaps a combination of particles in the environment, perhaps from work, perhaps from certain geographic regions, or perhaps an infection of some sort, exposure to a microorganism, a bacteria or a virus, can trigger the immune system to have an abnormal immune response. And that abnormal immune response involves different types of cells, including lymphocytes that produce cytokines. Cytokines are messenger proteins that can direct the fate of other cells and either promote inflammation and promote the formation of these well-organized structures called granulomas that are key in the pathogenesis of sarcoidosis, or they can direct their immune response elsewhere and resulting in non-persistence of these granulomas. And we know that these responses change the way that sarcoidosis behaves in the lungs. We think that that probably impacts the way sarcoidosis behaves in the brain. Specifically, cytokines have been implicated in the pathogenesis of sarcoidosis in general, include interferon gamma, TNF-alpha, IL-6, IL-12, IL-15, and GM-CSF. So these are a subset of proteins that are typically considered pro-inflammatory, and we'll see that they share a lot of commonalities of things that happen in infections, and specifically mycobacterial infections. So going back to our main question, which is, what are the biological triggers of sarcoidosis? What is happening to start that immune response? There's a lot of things that have been described, but not one has been proven as the culprit. We talk about a lot of occupational exposures, about genetic factors that likely predispose a person to react to that exposure in a specific way that promotes either autoimmunity or promote an abnormal immune response that causes the persistence of these granuloma structures. And really the thought about an infection being the trigger comes from the fact that sarcoidosis behaves very differently from other, let's say, autoimmune disorders. It's not that frequent to have persistent granulomatous inflammation that becomes chronic in this way. And that's really the pattern that we tend to see in fungal infections and in mycobacterial infections. Mycobacteria is the bigger family, and the most common infection there that people may have heard about is tuberculosis. And the fact that sarcoidosis clusters geographically, and what I mean by that is there are certain regions in the world where sarcoidosis is more common, but also it clusters within families. We know that in the same household, once somebody has sarcoidosis, other people are a little bit more likely to have sarcoidosis. So that supports that there may be something in the environment that triggers this immune response. And those two together start to suggest the possibility of an infectious trigger. Now, tuberculosis has come as a suspect, or the larger mycobacteria family, because tuberculosis and sarcoidosis share a lot of similarities. They are both granulomatous diseases, so they both cause this specific form of chronic inflammation called granulomas. They look very similar. They cause a lot of overlapping symptoms, usually weight loss, fatigue, night sweats, chronic cough, shortness of breath. When it affects the nervous system, which is my area, typically both can cause chronic headaches, problems with the nerves that move the eyes around or the face, the cranial nerves. And they actually, on imaging, when we do CAT scans, when we do MRIs of the brain and the spinal cord, they look strikingly similar in the same pattern affecting the base of the brain and the meninges called vascular meningitis. When we look at the immune system of both diseases, of patients with both diseases, they both have similar patterns of dysregulated T cell and macrophage responses. These are cells that are key in the maintenance and formation of those granulomas, and they tend to react in similar ways. While they're very similar, they're also different, right? So in one, there's an active infection with a lot more tissue destruction and what we call necrosis, which we can see in the pathology as caseating granulomas, which is that necrosis in the center of the granuloma, which is much more likely to happen in tuberculosis than in sarcoidosis. And of course, the treatment strategy is different. In one, you're using antimicrobials, and in the other one, we're trying to suppress that abnormal immune response. Yet they're similar enough that in order to diagnose somebody with neurosarcoidosis, we're obligated to check for tuberculosis, and oftentimes, even after a negative test, doubts remain, and we have to keep checking. And if the person doesn't respond to treatment, it may be that we missed tuberculosis in the first place, and we need to reassess. Now, this idea of mycobacteria or tuberculosis as a possible trigger of sarcoidosis is not mine, it's not new. This has been a topic of interest in sarcoidosis in general and in neurosarcoidosis for many years. Here are some old papers. I think the oldest one I found was in the 50s of observations of cases of Boeck's disease. That's a historic name for sarcoidosis, after the BCG vaccine. So the BCG vaccine is the tuberculosis vaccine that is sometimes used in some countries around the world. And there's been reports of sarcoidosis being triggered or having started after this vaccine, which suggests really that exposure to that to those antigens, to those proteins of the mycobacteria may actually trigger the disease. There are also reports of pieces of tuberculosis bacteria in some cases of patients with sarcoidosis. There's also evidence of mycobacterial DNA, so not necessarily the whole bacteria and an active infection, but remnants or evidence that the mycobacteria was there in patients with sarcoidosis. We've seen in studies, more recent studies, that looked at cells involved in the immune response of sarcoidosis, such as T cells, and how they react against different proteins. We've seen that there is a reactivity against specific proteins associated with tuberculosis, such as MCAT is one of the candidates that was explored by one of our colleagues, Dr. Chen at Hopkins. And multiple proteins. So when I say antigen is these proteins that cause an immune reaction. Multiple antigens of mycobacteria seem to be targets of those adaptive immune cells in pulmonary sarcoidosis. So while we've suspected this for a while, we haven't been able to prove a causal relationship. So we're not yet at a point where we can say, yes, mycobacteria is actually the cause of sarcoidosis, but there are some clues that suggest that it's part of the equation. So what we wanted to do and what we still want to do is to investigate further this link between exposure to mycobacteria species and the development of sarcoidosis, and specifically the development of neurosarcoidosis. So why study neurosarcoidosis specifically when we want to answer a question about sarcoidosis more in general? The reason is that sarcoidosis can affect many organs, but the nervous system is a little bit protected from the environment, is privileged. So we're not, when we look at the brain and the spinal cord, and we look specifically at spinal fluid, we have less noise, less contamination of exposures as compared to, for example, the lungs or the eyes or the skin that are the other more common organs affected in sarcoidosis. And there's also a relatively privileged immune situation. So we have less populations of immune cells in the spinal fluid. Typically, we don't have a lot of cells when we measure. So whenever we see something in the nervous system, we really think that that's indicative of pathology when we see increased cells or we see bacteria growing in a culture. So really, by going after the spinal fluid as a source of information, we can clarify a lot about not only neurosarcoidosis but sarcoidosis in general. So that's what we started to do. So this is showing first a study that was done by Dr. Chen years ago, looking at a protein called serum amyloid A. So amyloid A, serum amyloid A, is involved in tuberculosis pathogenesis. We've known that for many years. And what Dr. Chen demonstrated is that it's also involved in regulation of that inflammation of granulomas in sarcoidosis. This was done through an in vitro model of pulmonary sarcoidosis. So we knew that probably this protein has something to do in the development of the disease, or at least contributes to the maintenance of granulomas. So we wanted to take a look and see if we could replicate this in neurosarcoidosis specifically. And what I'm showing here is work done also in collaboration with Dr. Maria Reyes, who was at the lab at the time, showing levels of that soluble amyloid A across different groups, including healthy controls, other autoimmune disorders like multiple sclerosis, neuromyelitis optica, which are often confused or misdiagnosed, or common sources of confusion with neurosarcoidosis. We saw patients with systemic sarcoidosis, pulmonary sarcoidosis, for example, with lymphomas, here in the dark blue with neurosarcoidosis, and there with infectious causes of meningitis, neurosyphilis, and infectious meningitis. And what I'm showing here is that in neurosarcoidosis, specifically in the spinal fluid of neurosarcoidosis, we have these elevated levels of SAA in comparison to the other autoimmune disorders, but look at the infectious meningitis category. So it looks very similar to the neurosarcoidosis group. So likely this protein can help us differentiate, we think, neurosarcoidosis from other autoimmune disorders that may mimic neurosarcoidosis, but not necessarily from infectious meningitis, and certainly not from tuberculosis. But the fact that they look similar is suspicious, because it's behaving like tuberculosis does. Similarly, there's been interest recently in this other protein called soluble interleukin-2 receptor, which has been shown to be elevated in tuberculosis. So this is what we call an acute phase reactant. Also, it's a marker of lymphocyte activation. So whenever there is inflammation and the lymphocytes activate, they start pumping out this protein into the blood, and we can measure that. And people noticed a few years ago that this was elevated in systemic sarcoidosis in the blood. And then there's been interest to measure that in the spinal fluid, which has been done by a couple of groups now. And it's interesting because this protein has been shown to be elevated in the CSF of people with neurosarcoidosis. But if you look at this red box here, this is from that study. This is not our study. This is by Dr. Otto. The pattern is very similar to that of neurotuberculosis. So this is a marker that's similar to the other one that I just showed you, appears to be useful to differentiate neurosarcoidosis from other autoimmune disorders, but it's behaving on track or very similar to neurotuberculosis. This one specifically seems to also be elevated in CNS lymphoma, which makes sense because it's a marker of lymphocyte activation. So again, we don't have a specific biomarker, but the biomarkers that we use so far seem to be also elevated in mycobacterial infection of the central nervous system. Now, this is an experiment from our lab where we wanted to measure this IL-2 receptor. And again, we see here elevation in the purple box in neurosarcoidosis compared to other causes. So the NIC would be non-inflammatory controls, MS, systemic sarcoidosis. And then, while this is very promising, if we compare this with cryptococcal meningitis, so this is a common cause of infectious meningitis, and we had samples of this to compare, then this is very elevated in cryptococcal meningitis. So once again, the patterns that we're seeing in neurosarcoidosis seem to mirror what we see in infectious causes of meningitis and CNS disorders. We looked at other markers, specifically inflammatory cytokines, because we know from the studies done in systemic sarcoidosis that these specific cytokines have been linked to the development of sarcoidosis. So we wanted to see if they were elevated in neurosarcoidosis and if they could be potentially used to differentiate neurosarcoidosis from other causes. And we saw something similar. So TNF-alpha, which is the target of common treatments now used in sarcoidosis, the TNF-alpha inhibitors are blocking this signal here, things like Remicade, Humira, etc. And so we see elevations in the spinal fluid in this TNF-alpha protein as compared to systemic sarcoidosis, to MS, to NMO, and controls. Similarly, we see that with interferon gamma, with IL-6, and IP-10. So this is interesting because the thought now is that not one of these, but a subset of these in combination could potentially help us differentiate neurosarcoidosis from other causes. And when we try to do that, so what this is a plot of these proteins, these cytokines, so including the ones that we initially measured before and others, across categories, including neurosarcoidosis, other sarcoidosis in other organs, controls, and inflammatory diseases like MS and NMO. And we saw, that these clusters, meaning the samples with neurosarcoidosis appear to have the same pattern of signaling for some of these proteins, which are here. And then we started to think, okay, maybe there is a biomarker panel that we could use to diagnose or to support the diagnosis of neurosarcoidosis that can help us differentiate it from other disorders. The issue is that what happens when we try to differentiate neurosarcoidosis from infectious meningitis. So we looked at some of these proteins, that the inflammatory cytokines that had been identified in the prior experiment, and now we are using a different platform called Ella. So it's a similar but more accurate technology than we had used before. And here we see, yes, our cytokine panel seems relatively good at differentiating from other causes and non-inflammatory disorders. But once again, the patterns that we're seeing are very similar to what we see in infectious meningitis. So this is to say, with the cytokine signaling, that first, I think our main confusion factor when we try to diagnose somebody with neurosarcoidosis will be differentiate from tuberculosis and other causes of infectious meningitis, more so than differentiate from other autoimmune diseases like multiple sclerosis or lupus, for example. And it's very interesting that across different proteins, across different potential markers, what we're seeing is similarities between the behavior of neurosarcoidosis and the behavior of infectious diseases, which in my view at least support the idea that the biological process may be similar and perhaps that there is indeed an infectious trigger to neurosarcoidosis. Now, really the goal is to try to identify what that trigger is. And we started by doing an extensive literature review using publicly available databases of, and specifically this one is a database that is freely available, and it catalogs all the experimental data that we have on reactions of T cells, B cells, the MHC complex against different proteins. So with that information, we have identified so that there are proteins involved in the pathogenesis of tuberculosis and proteins that are involved in sarcoidosis or have been reported to be involved in sarcoidosis, and some of those overlap. So I put the intersection here in this table and highlighted in red the ones that are considered virulent factors or proteins in tuberculosis that contribute to the infection severity or the infection course. And interestingly, this overlap, contains a lot of those proteins, including some of the ones that we had seen before, like CAG. So this, to start, it really says, there's been at least 34 proteins that have been linked to both diseases, and perhaps one of these could be the trigger of that immune response. Specifically, there are some suspects. So MCAT has been described to be recognized by T cells in both sarcoidosis and tuberculosis. The mycobacterial heat shock proteins, so these are also some of these virulent factors in tuberculosis, have also been observed to be recognized in both conditions. And specifically, those seem to drive activation of those cells and cytokine production and contribute to granulomatous inflammation. And there are others, like the SAT family of proteins, that have been found in both tissue of sarcoidosis and tuberculosis. So we wanted to use new technology to try to sort this out. And there is this technology called FIPSIC that I'll explain in a second, that is relatively new and allows us to measure all the antibodies that a person has in their blood or in their spinal fluid against all the exposures they've had in their life. So it's pretty powerful. So what this is essentially is, instead of trying to check one-on-one, so one sample of one person against one protein that I think may be relevant, let's say a person with sarcoidosis against a test for the CAG protein, what this creates is a large array of proteins and protein pieces called peptides that we can then expose against many samples at the same time, such that we can identify if a group of patients or a group of people sharing a disease feature, let's say neurosarcoidosis versus tuberculosis, how those populations react against a large set of proteins. So this technology was developed by Dr. Benjamin Larman at Hopkins, and the way it works is that from the available databases, here UniProt is the one that people know. Essentially, we have access to now the genome, to the readouts of all the genetic material of many microorganisms. This is publicly available, and we can take that and break it into little pieces, and each little piece can be converted into a little piece of DNA. So we can actually manufacture the DNA that corresponds to a little piece of protein from the database. And what we can do is put these little pieces of DNA into viruses, and these viruses incorporate the DNA and start making the protein of interest, the little piece that you want to query. It's expressed in the surface of these viruses. And then at the end, what we have is a large array of these viruses called phages that express little pieces of protein, and we can take patient samples, and put it against this library, this large array, and then identify which samples reacted against which specific piece of a specific protein. We then sequence that and put that against the database of all the organisms that we can have, that was made to build the library. And at the end, what we get is signals of the patient samples reacting against a peptide that can tell us, that peptide belongs to that protein, which is from a specific species of interest. And in that way, we can capture in a broad manner the exposures that somebody has had against subsets of microorganisms of interest. So while that sounds very complex, it is. It's been in a way simplified by Dr. Larman's team. So they created one of these libraries called VirScan, and what that is a library of all viruses that have been described to infect humans. And they broke all of those viruses into little pieces, put all the pieces in this plate, and now we have a test where we can put a sample against this and see what immune response against any virus has somebody developed over their lifetime. So it's pretty interesting. And for example, this technology has been used recently to link the Epstein-Barr virus to the development of multiple sclerosis, which had been long suspected but not necessarily proven until this provided a little bit more evidence. In a similar way, Dr. Larman and his team have created a library, so similar concept to the VirScan, but instead of having proteins of viruses, created the human peptidome, so all the proteins expressed by humans, to check again for autoimmunity, for antigens that the immune system may mount abnormal responses against. So understanding that we wanted to create something like that, where we could query our hypothesis of mycobacteria being the trigger of sarcoidosis. And we tried this a few times during my fellowship. The initial attempts were not successful because it turns out that mycobacteria is, the genome of mycobacteria is significantly larger than the genome of a virus. So if we wanted to include all possible pieces of all possible proteins of all the mycobacteria species that exist, that was extremely several orders of magnitude larger than the VirScan library, for example, and it was just not feasible. So then we spent some time brainstorming, how can we reduce this in a way that's still meaningful for what we want to answer? And we landed in our database, the IEDB database, which, like I said, is a database that already has proteins that have been considered to be antigenic. So what that means is that from all of those possible pieces, this is a subset that already has been described or predicted to be potentially causal of an immune response to bind to a receptor of lymphocyte, T cell or B cell, or or been described in a paper. So this was a very good source of information. So we decided to focus on that. And what we did was try to build a mycobacteria library to try to characterize the mycobacteria exposures in sarcoidosis. So by using this database, we searched for all the peptides, all these protein species that were that belong to Mycobacteriaceae family, to not only tuberculosis but all related bacteria, and we came out with 1,565 antigens across multiple species, so much smaller but potentially more relevant. And what we did was, instead of stopping there, we went to our larger source called UniProt, where all of these other genomes of other bacteria are available, and we looked for similar proteins to the ones that we had found, such that if we had a little piece in the first database, we went to UniProt, looked for similar proteins, and expanded our library to capture not only the one that we had in the literature, but any other possible similar one, with capturing their differences across different species. Not limiting ourselves to tuberculosis, but expanding to all related species of Mycobacteriaceae. So at the end of that long process, summarized into slides, we ended up with 233,995 individual pieces of protein that we included in our library. And that represented over 200 species. And this is the list of the top 20 that were represented, with the amount of peptide tiles that is unique to that species. And of course, the largest contribution is Mycobacterium tuberculosis, which is the more common one. But as you can see, we have a broad representation across related species. And some of these mycobacteria that are listed there are environmental. So they are not thought to cause disease, but they may be in the water, they may be in dust. So it's interesting to see if we can capture if environmental exposures to mycobacteria, specifically non-TB mycobacteria, may be potentially a trigger of sarcoidosis. So after all of this, what we did was, once we had our list of proteins from the immunoepitope database, and we expanded it to homologous proteins in the larger UniProt database, and we had all of those list of proteins, we collapsed them such that to avoid redundancy. So if they were too similar, we only put the shared piece once, and then we capture the tiles or the pieces that were different, and we put those pieces of protein, chop them, and then made them into DNA pieces that, similar to what Dr. Larman had done with Verscan, these DNA pieces were then cloned or inserted into the genetic material of these viruses called phages. And then these phages, these viruses here, start showing up and expressing that protein. And now we have this big map of all of these little pieces of mycobacteria that we can test our samples against. And that's exactly what we started doing. So we put patient samples from different conditions, including neurosarcoidosis, systemic sarcoidosis, but also we wanted to test if this worked. Of course, so we wanted to put mycobacteria, proven mycobacteria infection, including tuberculosis and non-tuberculosis mycobacteria. And we got samples from a rabbit model with tuberculosis also as the positive control and healthy patients. And we put all of these samples against our library. And then the antibodies that we have from historic exposures, that is, against potentially this mycobacteria, would bind to this phage that is expressing the protein. And then we can precipitate these using something called magnetic beads. So essentially some magnet balls attached to the antibodies, and then we can pull them down to identify which ones reacted. And then we can amplify that sequence of DNA and reconstruct it. And with that analysis, we can say, okay, so in this group, there was a reaction against this specific piece. And then we need to figure out, that piece belongs to which protein and to which mycobacteria. But we're very excited because although up to here, I've only shown you the development of the library, this took a lot of work, and having a final list of mycobacterial peptides that is comprehensive and that we think is antigenic is is very exciting, and we hope that this can be used to really test this hypothesis moving forward. So we've done some preliminary experiments, once the library was done. And this is one of the first experiments we did, checking with in the library controls, non-TB mycobacteria, the rabbits that are the tuberculosis model, sarcoidosis, and human tuberculosis samples. And what I'll show you is the results for the rabbits. So each of these columns is a sample, an individual rabbit. And these are the ones with active tuberculosis. This is a control rabbit without infection. And these are rabbits that had tuberculosis, no longer have active tuberculosis, called convalescent. And the y-axis here is how strong, it's called a fold change, is how strong is the immune response against a certain tile, a certain piece of protein. And each little dot is a peptide, one of the reaction against that individual peptide and the maximum reaction that we have. So you can see that there is reactivity. So the line here starts at 5. So we consider anything below 5, we don't consider real reactivity. And you can see compared to the controls, which is relatively flat, that these rabbit samples have a lot of dots that are higher up in value than the other categories. So this was huge because this tells us that after all that work, the library works. So we got samples that we know should react against mycobacteria because these were truly infected with tuberculosis, and it seems like it's reacting. So great. And what we're seeing here is the list of proteins that have the largest reactivity. So they are ordered from highest to lowest maximum reactivity. The color helps. So red is more reactivity and green is less reactivity. Just to show that if we consider 5 the threshold, we have reactivities in the 500. So very strong reactivity for the active tuberculosis rabbits as compared to the control, for example. And interestingly, we don't really see much reactivity for the same proteins in the active tuberculosis than in the convalescent. So convalescent meaning they no longer have tuberculosis, but they were exposed. So here is, moving down that list, we see reactivities for these convalescent rabbits, but not for the active rabbits. So this is interesting to me because it tells me that the immune system has different reactivities for active mycobacteria infection than for a prior exposure, and they are reacting to different pieces in different proteins, which we can use to study sarcoidosis later.
Dr. Barreras, sorry to interrupt. I just 2 minutes left.
Okay. All right. Okay. So here, similarly to the rabbits, we took samples from humans with pulmonary tuberculosis, and we saw that there was reactivity against mycobacteria peptides in the library, which we're very excited about. So again, sort of proving the point that you not only detect reactivity in rabbits, now we're detecting reactivity in human tuberculosis, which is great. And similarly, we see here the list of proteins that have reactivity as compared to the controls, and it's very clear that we're detecting reactivity here. And then we started to do experiments now that we know it works with patients with sarcoidosis and other controls. So here we have the first 2 columns are controls. The middle column is the interesting one, is the sarcoidosis patients, and then we have tuberculosis and Crohn's disease, which is we used as another granulomatous disease to test. And what this is showing is that the reactivity clusters. So this is a list of proteins, and you can see in red that all the Crohn's sort of react in a similar pattern, and the TBs react in a different pattern, and sarcoidosis reacts in a different pattern. So we're seeing here that there is reactivity in the sarcoidosis samples against mycobacteria peptides that is different, or seems to be different, at least here, than the reactivity that we see in tuberculosis and the reactivity we see in Crohn's disease. So this is very exciting for us, and we need to then analyze, what these specific reactivities are and what's the relevance of that. And we need to do a lot more analysis before I can show you the final conclusion. But ultimately, some of the peptides that we're finding to be reactive in the sarcoidosis samples are peptides that have been linked to tuberculosis pathogenesis that include this PPE family of proteins that are, very well known to be antigenic in tuberculosis. So this is exciting, and I think there's a lot of work that we need to keep doing, including validating this with more experiments and continue to analyze specific candidate peptides across these groups. And ultimately, what I want to do is, once we have these immune responses, can we use that? Does that predict who gets a specific phenotype of sarcoidosis or who gets relapses? So we don't know yet, but more to come. So in conclusion, the clinical features and the pathological features of sarcoidosis and mycobacteria infection overlap. There are proteins such as cytokines and chemokines that are associated with both sarcoidosis pathogenesis and chronic meningitis and mycobacterial infections. FIPSIC technology, it's a great tool that can be used to screen prior and current exposures to microorganisms, and we can use this to investigate sarcoidosis. And we have developed now a platform to check mycobacteria peptides that seems to be reactive as expected with samples from a rabbit model of tuberculosis and human mycobacterial infection. And we have now seen sarcoidosis samples react against a subset of those peptides in a pattern that seems different from tuberculosis. So we need to figure out, what that pattern is and what's the role of that reactivity in the development of different phenotypes and disease activities. And like I said, this is a team effort. So all of these people have contributed to the data I showed you, and I'll take some questions. Sorry, I exceeded your 2 minutes, but hopefully not too much.
Fantastic. Thank you so much, Dr. Barreras, for your excellent presentation today. And we will go ahead and begin our Q&A at this time. As a reminder, if anyone has any additional questions, please do feel free to add those at the Q&A button at the bottom of the Zoom window. So Dr. Barreras, a few questions came in from the chat. The first one for you was asking about how and what about sarcoidosis allows it to cross the blood-brain barrier. So how do you think about sarcoidosis when, and its ability to cross the blood-brain barrier, and are there any considerations for this in your research?
Yeah, so that is a fascinating question because, of course, neurosarcoidosis is not the most common form of sarcoidosis, right? Like the most common form would be lung, skin, eyes, which are the organs that are in direct contact with the environment, and the CNS that is a little bit more secluded seems to be less affected. There was some interesting data that we published a few years ago in spinal cord sarcoidosis, where it seems like the areas of inflammation correlated with areas of disc disease. So whenever in the spine there was a disc sort of like irritating or indenting the meninges around the spine, sort of like disrupting that blood, in this case, a spinal cord barrier, it seems like there is where the disease, started and got established. Which may suggest that the disease starts in the outside and something else has to disrupt the brain-blood barrier to let it in. But that's still an area of active research.
Fantastic. Thank you so much. And second question we got from the chat, we had a question about competing diagnoses between neurosarcoidosis and specifically diabetic neuropathy. Do you have any insights on this?
That's a very difficult one clinically. So yes, so oftentimes we have a subset of symptoms that can be from one or the other, right? This is true for specifically in the case of small fiber neuropathy, for example, which is very common in patients with sarcoidosis. We know that sarcoidosis itself can cause a small fiber neuropathy, and we know that diabetes can cause a small fiber neuropathy. They can look very similar. And based on exam or the nerve conduction study, we cannot tell you which one is which. And that's where a biomarker would be useful, and we're not there yet. Eventually, when we're working on a similar study with the cytokines that I showed here, but in small fiber neuropathy. If we validate a subset of these markers that can tell me, more likely this is from the sarcoidosis than the diabetes, then eventually we can use that to justify treatment, with immune therapy. Because right now, we don't really use immune therapy for diabetes, for example. So it's very much a need and still a research question. But yes, tricky to figure out. Usually what we do now is try to control the sarcoidosis and the diabetes, but it gets tricky when the sarcoidosis seems stable and it's not obvious that we need to escalate immunotherapy sometimes.
Fantastic, thank you. We also had a question in the chat. Is it possible that these identified pathogens and patterns, similarities to other diseases, might suggest possible paths to treatments for neurosarcoidosis down the road?
Yes, but it's a little premature. So I think there are a couple of things that can come from this. First is if we truly identified a trigger and a cause, then we need to figure out what the source is, limited exposure, for example. I don't believe necessarily that there is an active infection in sarcoidosis and role for antibiotics, although some groups have suggested that. But it may be that there was an infection that came and went, and perhaps pieces of the microorganism stayed, and the body was not able to clear them, for example, or a certain aspect of the immune response overreacted to that infection in a specific way. So if we understand that piece better, perhaps, yes, we could target and limit that abnormal immune response.
Great, thank you. And then you were talking a little bit about some of the database and libraries that you had mentioned and used in your research, and they align with infectious exposures most often found, and are analyzed. Are those aligned with specific occupations or geographical locations? So, in other words, can these findings suggest risk factors for neurosarcoidosis? Maybe looking at geography, occupation where those infections might be found in the library.
So that's a fascinating question. We haven't done that test, but we could answer that question with this technology. Yeah. So perhaps for example, if we were to compare groups from different geographic regions and we were to see different exposures to specific mycobacteria peptides, then we could tell you, although probably the exposures are different depending on geographic region, if we find a commonality, maybe that's, a more relevant trigger. Or perhaps, if we find a specific mycobacteria, and then we go and look where is that specific mycobacteria, and we find that it correlates with specific regions where sarcoidosis happens, then that would be a more robust finding. We're not there yet, but we're definitely, going to do that.
Absolutely, fantastic. Great. And next question, would you be able to speak to the importance of incorporating, biomarker panels like you had discussed, incorporating those results in relation to clinical characteristics of a presenting patient? Can the biomarkers alone help to inform a diagnosis of neurosarcoidosis, or does it require kind of having that full context behind, the patient and their unique experience?
So the dream is to have a yes/no test for sarcoidosis, right? Eventually, we would want a subset of markers that together, ideally a one biomarker, but I don't think that's how it's going to look. It probably is going to be a subset of markers that we can send in a patient and say, your probability of this being sarcoidosis is 90%. That's the dream. The reality right now is that none of the markers we have, like the ACE or the IL-2 receptor, at least on their own, they're not good enough yet to behave as a yes/no test. So you do need this context, and specifically, probably these markers right now are very good or are better at differentiating neurosarcoidosis from other autoimmune disorders, but are not as good at differentiating neurosarcoidosis from infectious causes. And infectious causes are, a big mimicker of sarcoidosis, specifically tuberculosis and fungal infections. So I think we need better tools to differentiate those two.
Excellent, thank you. And we also had a question. Have you found any potential links to a risk of cancer in your neurosarcoidosis research? Is that also potentially one of those mimickers you have to watch for between neurosarcoidosis and cancer?
So yes. So the other big mimicker in neurology and pulmonology probably too, but in neurology is lymphoma. So lymphoma can affect the nervous system and can look just like sarcoidosis. And the tricky part is that some of our therapies for sarcoidosis may increase the risk of lymphoma a little bit because they are impacting lymphocyte pathways. Specifically, TNF-alpha inhibitors carry a little bit of increased risk of lymphoma, and sarcoidosis by itself has been described to carry a little bit increased risk of lymphoma. So anytime something like this develops, if we make a diagnosis of neurosarcoidosis, especially when it's done without a biopsy, which happens more commonly in neurosarcoidosis than in other areas. Sometimes we have, let's say, a biopsy of the skin or the lung saying that the person has sarcoidosis and just the brain looks compatible, so we don't go and biopsy the brain. In those cases, it can always be a lymphoma, and we always worry. And if the person does not respond to therapy as we expect or as they should, then at that point, we always should requery, are we missing a lymphoma? Yeah.
Thank you so much. And are you aware of any evidence that neurosarcoidosis might be caused by different pathogens than maybe the pathogens that cause the other manifestations of sarcoidosis and other organ involvement?
So there are a few suspects. The mycobacteria, I think, is the one with the most evidence. But the other one that people talk about a lot is P. acnes, which is a different bacteria that we often find pieces of in the biopsies that we do for sarcoidosis. So not uncommonly, we get a report that says non-Caesarean granulomas and P. acnes grew, and we're like, okay, is that an infection or do we need to treat this? The challenging part is that P. acnes is a normal bug that lives in the skin, so usually doesn't cause any problems. And because it lives in the skin, it's very easy to contaminate a sample. And if you're the physician, if you're the pathologist manipulating things, it can be a contaminant. But it's been reported enough times that there is suspicion that is beyond just a contaminant, that it may be a contributing factor, but that's not yet established.
Very great insights. Thank you. And general question. So what are you kind of planning next in terms of your research? Where would you take it from here?
So we're doing a few things. For this specific project, now we've done experiments with sarcoidosis samples, including cardiac, pulmonary, and neurosarcoidosis. I didn't show a lot of those results because they're too green to show. But now the experiment is done, which I'm excited about. So now we're trying to deconvolute all of the complex information. And we need to do a lot of bioinformatics to come back and tell you which is the culprit, if there is a culprit, antigen. So that will take some time, but the experiments are done. Is the analysis what follows? And after that, probably we need validation, with a different set of samples to see if we, replicate and see the same thing. And with the cytokine story, we're doing something similar, now outside Hopkins, but in collaboration with the small fiber neuropathy samples. So I'm querying to see if we can identify similar increases in subsets of cytokines in small fiber neuropathy, precisely because of that need that we talked about before of differentiating it from the diabetic or other things that may not be immune mediated. So if we see that a subset of markers help us identify who is truly from the sarcoidosis, then that would be helpful to guide who needs more therapy and who doesn't.
Excellent. And a final question for you this evening. Would you be able to speak to why this type of research funding can be so vital to helping to improve the care for patients that's available?
Happy to. I think this is critical, and I'm gonna say that this was key in my career. So at least in the neuro world, most programs that would train somebody that eventually sees neurosarcoidosis are MS focused, multiple sclerosis focused, and they're called MS fellowships. And there's really no program that is training people in neurosarcoidosis. So without FSR, I don't think I would have gotten the specific training that I got, and I don't know that I would have, been able to continue this interest and have now a dedicated clinic for neurosarcoidosis. And I know another, trainee that currently has a grant that's also being trained at UCSF, who's also, sharing this this path. And so it's critical because it's not only allowing us to do research and discoveries. Ultimately, the research project is the means by which we also get clinical training and imaging training and the interest in sarcoidosis, and allows young people to start a career and a lifetime of contributions to the field. So absolutely important, and please do support FSR.
Fantastic. Thank you, Dr. Barreras, and for all of the incredible work that you're doing to push neurosarcoidosis research forward. And we're so happy to support your work. We are out of time for the evening, but thank you again so much. And for answering all of our audience's questions, I'm going to go ahead and pass things over to Elise Hoover for our closing remarks today.
Yes, thank you, Dr. Barreras. That's a fantastic presentation. It's so great to hear about some of this work from our fellowship awardees. So, behalf on FSR and our community, we really appreciate your commitment to driving sarcoidosis research. So, thank you. And thank you to all of our attendees for joining us. Please stay tuned for more webinars and great programming. We have a great community newsletter that'll let you know of any upcoming events. So, please sign up and have a fantastic evening. Thanks, everyone.